It's a pleasure to have Marco with us. So Marco did his PhD at Wisconsin, and then as a chief scientist in Adamas, and he's one of the most skilled people in diamond chemistry. And so today he's going to tell us about diamond coatings and also about how to bring nanodiamonds into mice and some very interesting experiments that they've done at Adamas.
Yeah, all right. Thank you for a very nice introduction and just for putting this all together. It's kind of a nice, very good use of an afternoon. So the goal is to tell you about some recent work that we've been doing. The majority of this has all been funded by different NIH projects, which I want to acknowledge. And the goal is to talk about targeted nanodiamond for the detection of unpaired electrons, spins in biological systems. And so really, the idea is to bring together two interrelated topics. For me, it's going to be coming from a chemistry perspective. That's where my expertise lies. But we're going to be touching on going to the biological world as well as material sciences. So for targeting, we have some pretty well-established work targeting diamonds to cancer models within mice. And so this has been published, and we've been advancing on this for the past year or two. And then the idea is to couple this with really recent data that we've collected really over the past, basically until we were quarantined. And I think it'd be interesting to talk about some recent findings in trying to develop nanodiamond sensing technologies to the point where they can be used by any sort of biologist rather than specialized physicists in the field. And so the idea in the long term is to really couple these two concepts together such that when we have biological questions, we can get a particle where it needs to go and then learn something interesting about that environment. So briefly, for those that don't know, basically the chief chemist at Adamas Nanotechnologies, and we are a diamond-centered company. We are extremely research-based, and this is how we have many collaborations all over the world. We really focus on research. And our main goal is to adapt diamonds to a variety of technological sectors and really trying to get high-level applications. And so in the non-biological field, we're working with lubricants, drug delivery, very popular for creating diamond films, CVD films. We make a very good precursor, which allows for very smooth films that we have a lot of customers for. But the current focus today is really to talk about imaging and sensing. These are two interrelated fields that relate to properties of relate to the same properties of diamond, but they're used in different ways. So these come to luminescent centers in diamonds. So the idea is to take a diamond particle, mill it into a very, very small size, and then use native properties of diamond for imaging. So within the diamond lattice here, we have a network of carbon. Nitrogen is naturally present in a low amount in the diamond lattice. And when you have the appropriate coordination of nitrogen atoms and then a vacancy in the atom, and they're coordinated in such a way, fluorescence arises. So you shine light, and then you can get light back. And so in a biological setting, this is useful for reporting on biological analytes. So depending on the number of nitrogens, if we have 3 nitrogens or 2 nitrogens or 1 nitrogen coordinated around this vacancy, you'll actually get different colors. And so some of the recent advancements that we've done have been to expand upon the most popular center, the NV center, which is red, and to expand the color palette, which will be relevant a little bit later in the talk. Specifically, the 3 centers are N3, H3, and NV. And because these are fluorophores that are encapsulated within a diamond particle, a diamond is an extremely stable and robust material. And this means that they are truly infinitely photostable. This is the key value which they provide. So you can shine light with any intensity or length, and you're not going to get changes or any appreciable changes in the emission. Because diamond is it's very well tolerated biologically, it's biocompatible, we have multiple colors, which allows for multi-channel imaging. So while this is a very good feature, There are a lot of fluorophores on the market, such as Alexafluor or Genelia dyes, that offer stability which is good enough. So we really have a motivation to look beyond fluorescent imaging, to really an idea where you'd be able to have a particle in a cell, use the optical imaging to obtain location data, but then also use that imaging to obtain other parameters that are occurring in the cell, be it temperature, magnetic environment, pH, things of that nature. So the nitrogen vacancy center of the 3 luminescent centers here is the most well-studied. And primarily because it has an unpaired electron in its negative state, in its NV- state. And this electron is sensitive to both the diamond lattice as well as the external environment. And because fluorescence arises from this nitrogen vacancy center, there is a relation between the optical readout of this fluorescence and the particle's environment. And so this opens up possibilities that extend beyond simple biological imaging to a lot of Ashok's fantastic work with C13 hyperpolarization, and then many other works which just relate to different types of sensing. So one thing which we are interested primarily in is the detection of paramagnetic species, or species that have unpaired spin. The most common of which are reactive oxygen species. And so there are a variety of these variants, as well as nitrogen, reactive nitrogen species that have unpaired electrons. So these occur naturally throughout oxygen metabolism, but upon misregulation, they can be signs of and lead to oxidative stress. And so detecting these different species within a cell in response to some sort of activated stimulus is of interest. There are also a number of other paramagnetic species within the body. For example, iron in blood is either paramagnetic or diamagnetic, depending on whether it's oxygenated or non-oxygenated. And so there are some pathologies which could be interesting to study optically, which because there are differences between these two states which are difficult to probe in whole blood applications. So using detection in these systems requires primarily high levels of sensitivity, so being able to detect low levels of an analyte, and then being able to localize the particle to an area where you can actually detect something. And these are the two main topics here of discussion. So first, I want to recount some work that we did targeting diamond to tumors in mice. And so the actual experimental design is fairly straightforward. When you have a tumor in an animal, it starts to grow. At some point, this excessive growth becomes starved for oxygen and nutrients. And at that point, the tumor starts to emit a chemical signal which causes blood vessels to grow. And that allows them to eat and breathe and continue living. So because this is a mechanism which is conserved throughout most, essentially all cancerous growth, we thought that this was a good target for trying to localize a diamond particle to a tumorous cell. So here you can see that as you start to get more and more growth of these yellow cancerous cells, you begin to get expression. Well, you basically get vascular genesis. Sorry, some heart here doesn't exactly look right. As you get an increase of these signalings to grow blood vessels, you'll get an increase of a particular protein called vascular endothelial growth factor receptor. So vascular endothelial growth factor is the factor which causes the growth of these blood vessels. And as this event is occurring, these receptors become upregulated. So there is a higher level of expression of these in tumors. So we hypothesized that it would be a reasonable idea to conjugate VEGF molecule to the diamond surface and use that to colocalize to cells that are overexpressing VEGFR2, which will be present in tumors. So in trying to accomplish this, we had to look at our toolbox of different chemistries and figure out what is the best way to affect conjugation and modification so that we can perform our targeting. Now, there are two main issues, one of which is that the particles need to be colloidally stable. They can't collect together in the way that nanoparticles like to do, such that they settle out and change in their sizes. And they also need to have carefully selected attachment of the targeting ligand. So we focused on a coating called polyglycerol, and you can see here in the structure, it's essentially a polyether with a high abundance of hydroxyls. So actually this hyperbranch structure provides essentially one of the highest carbon to hydroxyl ratios that is possible to achieve. And so this coating becomes very well hydrated, and that keeps the particles stable in high salinity environments, such as blood, where most particles would tend to aggregate. This is very similar to polyethylene glycol, so PEG coatings, which are commonly used. I would say this is perhaps a better variant. So one thing which I had to do, is figure out a way to get VEGF conjugated, or VEGF or some other target protein conjugated to the surface. To do this, we developed a variant of click chemistry, so something which is fast, doesn't require reagents to be used, and is transferable to multiple reagents. And so we settled upon this transcyclooctane tetrazine chemistry. So there was a lot of work in taking a tetrazine molecule and conjugating it to the surface, and then using that, as you can see here, to then conjugate VEGF to the surface. Now, a reason that this particular design was selected is that if you were to use standard NHS coupling chemistries where you randomly couple to any sort of amine which is on the protein, this VEGF molecule would conjugate to the particle in any variety of directions. Because we have a selective ligand, the VEGF molecules all bind to the particle in the same direction, and that increases the percentage, or increases the likelihood that the correct orientation is being expressed for targeting to the cells. And this is important for conjugation of valuable analytes such as VEGF. So some things I won't go into is that after this was done, we had to do a number of lab-based demonstrations. So SDS page and ELISA assays, and then functional assays to ensure that we had loading of VEGF on the particle, and then that the particles were actually functioning. So they were binding to VEGFR2 receptor, starting some sort of response. And then before we put them in animals, we wanted to make sure that there was no cytotoxicity. So once we had all of that,
so Marco, quick question. What is the coverage of the polyglycerol and the nanodiamond, and how many of these receptor molecules are you able to bind on average? And also, what is the particle size that you chose in these experiments?
So admittedly, it's hard for me to express a density per se. The layer thickness can be varied somewhat. It tends to add from 10 to 15 nanometers to more, depending on the reaction conditions. But on average, the layer is about 10 nanometers thick, with a very high density of hydroxyls. And I don't know that number off the top of my head. Similarly, the density of VEGF can be tuned depending on the number of, I go back here, depending on the number of tetrazines put on the surface. And this can be controlled anywhere from 2 to 3, or like say 1 to 5 per particle upwards of 400 to 500 per particle for 100 nanometer particles. There is a loss in stability once you get above 100 of these per particles, because you start to induce character which causes aggregation. So for a lot of these studies, the density of VEGF was in the realm of 100 ligands or so per particle.
So what are the sizes of particles?
So the majority of work which we did was with 100 nanometer particles. We did some exploratory work with different sizes. 100 nanometers is, well, the core sizes are, actually in this case, I think we're 140, which once they're functionalized and they have the proteins on them, they get closer to 170 nanometers, which is a good size to maintain retention during circulation, so that you can get higher degree of particles to your tumor.
I see. Is there any specific reason that you chose polyglycerol instead of PEG?
A couple reasons. One of which has to do with I found it easier to synthesize, and quite frankly, you get a higher hydroxyl ratio, which imparts better overall colloidal stability. If you look in the diamond literature for nanodiamond particles, you'll see a lot of functionalization with PEG, PEG. In some cases, you will see stability in biological media such as PBS or any sort of high saline environment, but a lot of times you don't. And that's because the density of the PEG is not sufficient to impart that colloidal stability. So the polyglycerol, PG, provides you with a higher density of hydroxyls and higher level of hydration, which then allows the particle to be resistant. I've done tests 4 molar sodium chloride or 3 molar sodium chloride, and the particles are perfectly fine.
Thank you. Thank you.
Yeah, and at any time, yeah, I'm glad. If anything is unclear, yell out. And I like this discussion is a fantastic idea. So after we had established that there was no toxicity, we went to cell targeting studies. In vivo experiments are costly in terms of each animal life, as well as the time involved in growing tumors and running the experiments. So we wanted to do at least some in vitro experiments first. And so here, this is the polyglycerol control, incubated with human endothelial colony-forming cells. And when you compare them to the VEGF functionalized cells, you see a huge increase, or you see a very strong colocalization with the cells, whereas you don't with the polyglycerol. Moreover, we compared these endothelial colony-forming cells, which express a high number of these receptors, with fibroblasts, which have roughly half, or I don't know if it's exactly half, but they have a lower number of VEGFR2 receptors. And then we were able to see that the number of particles bound under similar conditions correlated to the number of receptors. So we felt very confident in our in vitro studies that the targeting was effective and that it wasn't some sort of size effect or non-specific effect. So we moved to in vivo studies. And so to run you through how this works, we have a nude mice. And so we have my wonderful collaborator, Ashlyn, in the polymer group at Duke Medical Center. Imparts an orthotopic model. So this is a mouse model, but we're going to use a human cell line. Well, actually, sorry, it is a mouse line, but it is not grown in the same tissue. 41 carcinoma cells are then grown in the leg. And this is to allow us to have the best external imaging capabilities. I'm not going to go into it too much, but in addition, one goal here was to try full body imaging. And so we started with nude mice, which don't have hair, which allows for better visualization, as well as growing things in the flank are very reliable. They're very easy to grow in the eyes of our collaborators. The particles are then injected via tail chain, which allows for a systemic introduction. The mice are left for 3 days, during which we did do whole body imaging throughout. And afterwards, we excised the tissue, sliced them in a cryotome, and then did microscopy. So the results here...
How much particles were injected?
So in these cases, I believe it was 2 milligrams per mouse.
2 milligrams.
Yes.
So eventually, what would be some sort of a concentration that would accumulate at this tumor tissue? Is there any estimation of this?
So so that's a good question. We did not do dose response curves for all of the different organisms. What I can maybe show you here. So in this case, it's not. It is not strictly. This may or may not be directly related to your question, because these were untargeted, and we were studying something different here. Majority of uptake occurs in the tumor, spleen, and liver, and I don't have a solid answer. Probably anywhere from a quarter of the dose, probably about a quarter of the dose. There are some studies which have been done recently looking at accumulation of diamond particles in mice, which I could refer you to later. But this is an example. So we did do organ analysis, looking at least the fluorescence. One thing which we were interested in doing, if we are able to do a similar study again, is to actually then dissolve the tissue in acids and then recover the diamond to get some sort of mass-based answer. But at least through fluorescence, we can get a rough idea.
Okay, what's the control in that plot, please? Is that without tumor or without diamonds or something else?
So actually, this right here is just fluorescence. So technically speaking, they could be divided or subtracted. The control is just the autofluorescence of the tumor itself.
Okay.
And so we really only see signal from the tumor, spleen, and liver in these cases. And I think kidneys was also significant.
Did you measure the circulation time in the blood, or the half-life?
We were not able to reliably measure that in this study. We were hoping to get an idea of that via our time-dependent time-dependent full-body imaging, but the contrast was just not great enough. The best we were able to do was using 170 nanometer particles, which are a little bit larger than what was used in this study. It was part of this study, but a separate experiment, in which if the skin was removed, we could start to see accumulation in the organs. So this was modeling like a fluorescence guided surgery concept. But the inherent brightness of the particles is just not sufficient, and with the penetration depths at the maximum of 650 is just for the full body imaging. So we couldn't really do that.
I'm sorry, I'm not sure if I fully understood that image. So does that mean like the most of the diamond are kind of distributed all over the body, but that the tumor has only slightly higher kind of fluorescence than other places?
So that was a non-targeted example, and in that case, we were using a different size to exploit a non-specific targeting. Well, it is a phenomenon known as the EPR effect, enhanced permeation and retention. And in that case, there was an uptick in the tumor as well as other organs. The point of that graph was just to show which organs show the highest level of uptake, and that corresponds with general understanding of organismal uptake. When you're above 5 nanometers, you can't be cleared, so the particles are going to end up somewhere. So they're either going to end up in the kidneys or the liver, or the spleen is a huge one for uptake. But you do pose a good point. So there's a recent workout that does show that there is a lot of depositions in the skin and as well as in tissue. So if you do an organismal workup, organ by organ, you don't necessarily recover 100% of the particles. So that is an important question to address for applications where you are systematically, for like human use, for example.
Interesting. So, sorry, another question was that, so that was a 2D image. So, were you able to sort of characterize the 3D kind of feature or profile, like how deep can you image, and like?
It's a very good question. So I can. I wonder if I have present. I don't have them quickly accessible, but we did do larger, at least larger two-dimensional sections. So in terms of the 2D sections, what we noticed is that you have higher accumulation of the particles on the periphery of the tumor, where we think that you have larger blood vessels and a better vascular network. As you get deeper and deeper into the tumor, the particle densities become less and less and less. So really, so this is actually at the edge of where we get the highest densities.
Yeah. I'm just saying, like, was this fluorescence, so or kind of imaging technique? How deep can you actually image, or what is kind of penetration depths?
So in confocal, the penetration depths are less than a micron. Scattering very quickly becomes an issue. With multi-photon techniques, you can increase this. But these sections were 5 micron sections.
I see.
Yeah, that's helps. We have done some confocal in cells, but yeah, in general, really, even with a good confocal microscope, I don't know, maybe 20 microns. This is a good microscopy question to check up on. But so when we did the analysis of tumors, so control is just saline was injected to the mouse. So we're looking at tissue, non-targeted and targeted, and there was a clear increase in the tumor, which was significant. There was some marked increase by the PG. Hard analysis of this shows that there are some very, very small particles that do make it, but in general, they're difficult to detect. What was exciting about this was that we were able to take spectra to unambiguously confirm that what we were seeing was diamond and not some other biological effect, because there is a lot of autofluorescence which occurs under the microscope. You can see that there's a lot of red background here. Spectroscopically, we could determine that they were diamond based off of the signature. So that was very encouraging, and showed that at least in vivo models we could target to specific organelles, and we're hoping that more advanced versions of this could be performed based off of things that we've learned. So that was the bulk of the targeting work. And so I want to switch over now to the using of diamond in a sensing realm.
Marco, I'm just going to briefly just follow up. You mentioned this was showing targeting of organelles.
Organs, sorry.
Okay, great. Okay, because these are presumably cell surface expressed markers. Yeah.
Yeah. And so in this case, it's possible. So these VEGFR is internalized upon binding, but it's not clear that that is or is not happening. So future ongoing work with regards to targeting is actually to use smaller particles, which can then hopefully be targeted to like the mitochondria or specific organelles.
Cool. It'd be good to follow up with you maybe after the call if you don't mind.
Oh, yeah, that would be wonderful. Yeah, that's something that we're super interested in.
Great.
Can I ask, do you think you're targeting VEGF on the surface of an endothelial cell on a blood vessel, or do you think you're actually extravasating and going to a cell surface, a cancer cell surface?
I just don't know. I suspect, I don't know. I suspect that we are targeting areas of increased vasculature growth. So I wish that we could have done a co-stain of the vasculature to better elucidate that. That would have been more clear. I suspect that it is the higher level of vasculature, which is correlated to the tumor growth. Because it's such an active area of growth, you have a concomitant increase of those receptors.
I would call that a feature, not a bug, because you don't have to have the EPR effect.
Oh, well, yeah. Yeah, that would be my based understanding. Yeah. So one thing which I haven't talked about too much, but it's really relevant to this work and in our efforts to improve the capability of diamond as sensors is really what material are we using? So to make these different sensors, we're actually using some processes that were developed, really pioneered by Alexander Zaitsev at College University of New York, at being able to have a specialized furnace that brings particles up to a very high temperature very quickly and then back down very quickly. So typically the way that these luminescent centers are made is that the particles are irradiated with high energy particles to make these holes, and then they're heated to create vacancy centers. Now, all the particles that you find in the world, NV centers, are made by a pretty standardized process. Because you have this very long dwell time, you basically get trapped in this NV minus state, and then if you get too high, you induce graphitization. By carefully controlling this RTA process, you're able to tune the luminescent properties of diamond, creating a mixture of these three different color centers. And so this is absolutely Olga's expertise, and so I hesitate to say too much more at the expense of making a mistake. But the interesting thing, in collaboration with Ashok, is that these treatment processes seem to improve a lot of these interesting diamond properties. So hyperpolarization, the ability to optically polarize diamond spin, is dramatically increased. And so there are a lot of interesting applications that Ashok is looking at for using these as both MRI contrast agents or using them to transfer polarization, which would be really, really fantastic. But based off of those observations, we had a hypothesis that RTA would improve the degree of this modulation phenomena that I've been talking about. So we have a cast of players here. And sorry, yeah. So this is right. So sorry, Olga, that I forgot. The standard treatment is 2 hours for 850. So this is how the majority of all standardized NV particles are made, and you have this characteristic spectrum. Then we have RTA treated samples, or high temperature treated. So you see 1500, 1700, and 1900 for very short periods of time. And you see here at like 520, this peak growing in is more and more of the H3 center. And so we studied these systematic samples, and then another sample that we had a hunch might also be very interesting. And so here you can see what they look like under the microscope. And so here are these samples, and you'll notice that as the temperature increases, or the integration of the temperature and the time, this on-off difference begins to increase dramatically in a significant manner. Interestingly, in work that has been done with Ashok, the reason we selected this 1740 8-minute sample was that it had performed the best for particles that had been irradiated at a very specific fluence. And there's an interesting correlation between an enhancement of these quantum properties. So with the help of Alexander Shamas in Israel, we looked at some EPR data to try to understand what is governing this. And so it seems that an increase in T1 times and a lower NV content, as you see here, at the 1900, you have a much lower degree of NV minus, seem to be correlated with these, say, increased ability to respond to a magnetic field. I just want to thank lots and lots and lots of people that helped with different types of measurements, all the way from the hard physics to dissecting mice. So I was in the lab when helping. We were cutting out organs, and it's lots of different worlds, which is very cool. But it takes lots of really smart people to figure this out. And then NIH, has been really supportive in this work. So I really want to thank them. So yeah, thank you for your attention. Hopefully there's some good discussion that can be had, or